All right, welcome back to another explainer. Today, we're demystifying the four main types of IV fluids and looking exactly at how they interact with our body at the cellular level. We're going to take a fascinating microscopic journey to break down these vital medical fluids into crystal clear categories, so let's get right into it. Here is our quick roadmap for today. We'll start with the basics of osmolarity, then move through hypotonic, isotonic, and hypertonic solutions, and we'll wrap the whole thing up with those large molecules known as colloids. Starting off with Section 1, the basics of osmolarity. So before we look at the specific fluids, we absolutely have to establish the underlying biological rule governing all of them. Osmolarity is basically just the concentration of a fluid compared directly to your blood. It's this exact concentration that dictates the rules of engagement, right? It decides if water is going to move into a cell, move out of it, or just stay perfectly put. It is literally a constant microscopic battle for water balance across that cellular membrane. Which brings us to Section 2, hypotonic solutions and swelling the cell. The easiest way to remember how this works is just looking at the prefix. Hypo rhymes with low, meaning these fluids have a lower osmolarity or a lower concentration than your actual blood. And this low concentration acts as a massive trigger in the body. Because of that low osmolarity, the water is forced to move. It leaves the outside extracellular space and travels straight to the inside of the cell, into the intracellular space. The water is just desperately trying to balance out those concentrations. So, what are we actually looking at in a clinical setting? Well, the common types of hypotonic fluids you're going to encounter on a medical chart include half-normal saline, which is 0.45%, quarter-normal saline at 0.25%, and one-third normal saline at 0.33%. Now, here is the absolutely crucial point, the clinical risk. When you push all that water directly into the intracellular space, the cell swells up. Think of a water balloon filling up way too fast. If that cell gets overloaded with fluid, it can literally rupture. It's a dramatic biological event, and it is exactly why these fluids have to be so carefully managed. All right, shifting gears to Section 3, isotonic fluids, the perfect balance. So the prefix iso means equal. Isotonic fluids have an osmolarity that is perfectly equal to your blood. And because those concentrations match up perfectly, water transfers equally in and out of the cell. It's an even trade. This means we can effectively expand the intravascular space, giving the patient that much-needed volume without totally disrupting the delicate cellular balance. The cells don't swell, and they don't shrink. The heavy hitters you'll see here are lactated ringers, often just called LR, and good old normal saline. Now, what's absolutely critical to lock in your memory is that normal saline is the one and only solution approved for use alongside blood transfusions. Seriously, if you're giving a patient blood, normal saline is your one and only isotonic partner. But there's always a rule breaker, right? Watch out for 5% dextrose in water, or D5W. It actually enters the body starting out as an isotonic fluid. But once it's inside, the body rapidly metabolizes that dextrose. And then step 2 kicks in, and it actually turns into a hypotonic fluid. It's a total shapeshifter. Now, you might think equal means entirely safe, but that's definitely not the case. The primary risk with isotonic fluids is simply fluid volume overload, just giving the body way too much volume, way too quickly. Beyond that, because our shapeshifter D5W contains dextrose, it can cause an unwanted spike in glucose levels. And lactated ringers, that can be really dangerous for patients with liver disease. Why? Because a compromised liver just cannot convert the lactate in that fluid into bicarbonate, which leads to a whole host of further complications. Moving right along to Section 4, hypertonic fluids, or shrinking the cell. Okay, so we know hypo pushed water in. Well, hyper means high. Hypertonic fluids have a higher osmolarity, a much higher concentration than blood. Because the fluid outside the cell is so incredibly concentrated, it aggressively pulls water out of the intracellular space to try and dilute it down. This high concentration category includes some pretty robust heavy-duty solutions, like 3% saline, which is a massive jump from normal saline. It also includes dense dextrose combinations, like dextrose 10% in water, dextrose 5% in normal saline, and dextrose 5% in half-normal saline. And the dramatic medical risk here is the exact opposite of our swelling water balloon. By pulling all of that water out into the extracellular space, hypertonic fluids aggressively dehydrate the cell itself. The cell completely loses its structural volume and shrinks down to something really, really small, almost like a little raisin. So this brings up a really fascinating physiological puzzle. Up until now, we've been talking about water moving freely back and forth based on concentration. But what happens if the fluid molecules we're injecting are just simply too big to cross the capillary wall at all? Well, the answer lies in section 5, colloids, the large molecules. These guys operate on a completely different mechanical level. Because they're made up of very large molecules, these heavy lifters never actually cross the capillary wall. Instead, they stay trapped right there within the intravascular space, acting kind of like a massive sponge. They generate this really strong pulling force called oncotic pressure, which vigorously pulls water in from the surrounding interstitial space. So this unique group definitely isn't your standard salty or sugary water. We're talking about large, complex molecules like albumin, dextrin, and hydroxyethyl starches. And, super importantly, this category includes essential blood products like plasma, platelets, and whole blood itself. But because of their powerful pulling nature and their complex biological contents, colloids carry some very distinct risks. I mean, they aren't just shifting water around. They can actually provoke systemic allergic reactions. They can also rapidly cause fluid volume overload because of that massive oncotic pull. And especially when you're dealing with certain starches and blood products, they can introduce severe bleeding problems. Okay, let's summarize this entire cellular journey. If we map these 4 fluids directly against their cellular behaviors and risks, we see that hypotonic pushes water in, which risks rupture. Isotonic equals everything out, risking fluid overload. Hypertonic aggressively pulls water out, risking cell shrinkage. And finally, colloids stay put in the intravascular space, pulling water via oncotic pressure, bringing along risks of allergic reactions and bleeding. It's actually incredibly elegant when you view the whole shebang laid out like this. As we wrap up this explainer, I want to leave you with this final thought to ponder. We've just seen how very specific concentrations of medical fluids can force our microscopic cells to rapidly expand, violently shrink, or achieve this perfect, delicate balance. It really kind of makes you wonder, without an IV bag stepping in, just how incredibly precise and how fragile is the everyday baseline of our own human biology. Thanks so much for hanging out and learning with me today. Keep asking questions, and I'll catch you on the next explainer.